Thank you very much for the introduction, and thank you for joining my presentation. So again, today I'm going to talk to you a bit about the project that I started a couple of years ago. And as was mentioned by the first speaker, this was a resubmission. So we were funded not on the first attempt, but on the second attempt. And I can pinpoint some of the changes that we actually made in the project. So the specific platform that we're interested in is genetic sequencing. So genetic sequencing has a role, many different roles in public health. And I've described some of these. So like looking at paternity, looking at ancestry, looking even at forensic science. And one specific, oops, okay, is it going automatic? I don't think so. One specific thing that I'm interested in is identification of antimicrobial resistance or specific genes or specific microbial within a sample. So sequencing today has changed dramatically, but it can still be broken down into these four fundamental steps. So the first step is to identify a biological sample. So this could be a nasal swab, this could be a soil sample, this could be a blood sample. And then all the DNA or all the cells from the sample are extracted, and then the DNA is then extracted and then fragmented. This step is predominantly done in the lab. And then the sample is then transferred, or typically it used to be that the sample is then transferred to a sequencing facility where you have large sequencing machines where the sequencing is then performed. So you're simply reading off the DNA or the nucleotide sequence corresponding to each one of the fragments. So traditionally, Illumina sequencing was used, and it's still very frequently used today. The next step is then to do some initial steps in the analysis of the sequence reads. And then the last step is then to do final analysis and answer some sort of biological question. So the actual sequencing machines have changed dramatically in a fairly short period of time. So in the 1990s and even earlier, you required essentially a whole room or even a whole, I want to say, floor in order to perform sequencing. And it would take days, if not months. In the first human genome sequencing experiment, it was believed, or the folk story was that they hired undergraduates to actually read off the nucleotide sequence from the gels. So it required huge workforce and a huge amount of resources. Then between around 2000 and 2005, this process was largely automated. So the challenge was, how do you perform the same steps that all these individuals did for fragments in a high throughput manner that is doing multiple fragments at the same time without actually moving the sequencing, the sample? How do you perform the same steps in place? So there was a significant amount of breakthroughs. PacBio and Illumina came out with their technology, and it was picked up very frequently. It was picked up immediately, and it's still widely used today. Since around 2016, sequencing machines then became portable. So Oxford Nanopore came out with a miniaturized sequencing machine where it can plug in directly to a portable device, and it can perform the sequencing and produce the data from a biological sample in around less than an hour. So the amount of DNA that you can be sequenced when it first came out was rather small. Now you can do it, supposedly you can do a whole genome. but it was really targeted at microbial samples. So identification of like viruses, and in my specific research area, it became widely used because the question was, can you identify antimicrobial resistance via identification of a specific genes from a metagenomic sample? So portable sequencing, so Oxford Nanopore and portable sequencing would be extremely suitable in environments where you don't have accessibility to like a large computing resources and where a large sequencing lab. So one of the focuses that we actually looked at is in remote areas in the United States. So when we first proposed this, we described that there was areas where you're not necessarily guaranteed to have a high throughput, like a advanced network connection. Now, why that is needed is because at the time, and currently even today, the data has to be actually transferred to a cloud computing resource, predominantly Oxford Nanopore's proprietary service, in order to read off the nucleotide sequences, that is to do base calling, and then to do some sort of analysis. So basic analysis, including assembly and also analysis. This was all done in the cloud. So to us, this completely defeats the purpose of having these portable sequencers, where in order, you can sequence portability, you can sequence in a portable manner, but then in order to actually perform the analysis, you have to have a large-scale computing resource. So one of the things so this was not going to be suitable in remote areas of the United States. So I mentioned that this was a resubmission. The first time that we actually described this, where there was remote areas in the United States where you were not guaranteed to have a strong network connection with high throughput, reviewers came back and said that they didn't believe that was. So we actually had to, and I'm glad the panelist spoke in front of me, because he says we had to like tighten our claims and back it up with specific data. So we actually went to the FTC and collected data, a map of the United States where there was not like a strong network connection. And it was actually surprising. It was surprising to me. This was pre-COVID, where there was plenty of areas in the United States where there was not this network connection, and you would not be able to perform the analysis in this manner. So I think we had to do two specific things in our revision, which was to tighten our description as to what we were attempting on doing, and to back it up with specific data, to dispute any claims that, oh no, everybody has a strong network connection. And after that, we were funded on the second round. So our goal then is quite simple. It's to do all the analysis on a portable device. So use the memory, restrict interest to just having the memory and the CPUs used on a phone or a tablet to do the alignment, to do the base calling, do the alignment, and to deliver a result about the sample just using the portable device. So this requires sort of a transformation of the analysis. So at this specific time, the data was also getting larger. So, you know, you're able to analyze and produce data for viruses, bacteria, and then as we were actually preparing the proposal, there was a paper that came out that stated that you can sequence a whole human genome using Oxford nanopore. So in order to attempt on analyzing this data, we had to combine certain ideas, certain computational approaches. So this is also where my collaboration with people in other research areas came into play. So I teamed up with somebody that works in epidemiology and machine learning. I myself don't consider myself to be a machine learning person, but I work on succinct data structures. So succinct data structure reduces the memory, but still allows for efficient performance. So the question then became, how do we combine machine learning with these succinct data structures in order to enable the analysis to be done on the device? And that was kind of the true like computational approaches in order to deliver the results of achieving portability. So behind the biological problem or the informatics problem was a fairly deep computational approach. So succinct data structures and applying to succinct data structures to machine learning, there's some like fundamental problems there that we discussed when we prepared the proposal. And not only that, but there was some other incorporated into that is also looking at hardware and usability. So what we wanted to actually deliver was not just some algorithms, but we wanted a holistic solution that would enable somebody to go to a remote area of the United States and identify antimicrobial resistant bacteria. That was our goal. So in order to do that, we had to have a very collaborative investigative team. So I currently lead the project, but I work very closely with Casey Zheng, who is a biologist, microbiologist. He specializes in antimicrobial resistance. Then Matteo Prosperi, who I mentioned previously, he is in the Department of Epidemiology and focuses on applying machine learning to epidemiological questions and problems. Jaime Reese, who works on usability and HCI, and is also an expert in the analysis of or mobile analysis and portable devices. So that was our team. I should mention here, as the previous speaker said, is that when we were writing the proposal, this wasn't a new collaboration. So I had research papers and collaborations with each of these individuals far before we even thought of the project. So this is really this wasn't a new collaboration, but it was a ongoing collaboration that we could bring together. Yep. And that's all I have to mention.